Hello, Assalamu alaikum. In this video, we will understand about the ubiquitination. So, this process basically involve the attachment of ubiquitin molecule, small protein tag, small protein tag to target the proteins, so which that is unrequired for our body, for our cell. So it requires the enzymes such as enzyme E1, E2 and E3. So remember about that thing. Basically our unrequired protein will be recycled and reuse that amino acid and peptides for the another function. So here is the ubiquitin molecule, the protein containing molecule, combined with the enzyme 1 and substrate, with then enzyme 3 combined with the substrate to change their conformation and the degradation. But anyhow, let's understand about this ligand, basically the signal to the cell. So this is the person contains cell. So whenever this ligand will give the signal to the nucleus, so the transcription factor will begin the transcription of the DNA into the mRNA. This messenger RNA is ready to translation to synthesis certain type of the protein whenever the transfer RNA will pick up the amino acid to polypeptide formation, and this protein modification by this mechanism like the Golgi bodies, etc., etc. So in this way the ubiquitination will occur if the modification required, or the deubiquitination will occur also for the modification, but the degradation also involve for the ubiquitination to degrade the certain type of the protein which that can be cancerous or some other purpose like the unrequired protein. So in this way the ubiquitination and deubiquitination we will discuss in this diagram and in illustration. So in this way the ubiquitin first of all activate. The activation required the enzyme 1 and the ATP. The ATP basically will convert into the AMP, and the 2-phosphate will require, so it means the rich amount of the energy which that required for the combine the enzyme 1 for the activation of the ubiquitination. Then the conjugation is required. The meaning of the conjugation mean the enzyme 2 will combine together with the ubiquitin. So in this way the ubiquitin will combine as a conjugation, and in this way remember the enzyme 1 again use, so the reusable enzyme will recycle this enzyme to activate the further ubiquitin, and in this way the chain reaction will begin. The ubiquitin combine with the enzyme 2, and in this way it will remove whenever the enzyme when enzyme 3 embrace with the substrate to give the ligase enzyme to combine with the ubiquitin. So in this way, again and again its cycle lead to ubiquitin attached with the lysine site of the substrate protein. This protein is basically not required, suppose. So this is the activation, the conjugation and the ligation which that require for the again and again to combine the ubiquitin chain for proteolysis. So in this way, let's understand about the how proteolysis will occur. So the ATP again used for the proteasome formation. So the proteasome formation will occur. The 26 Swartberg unit containing proteasome. This is the 26 Swartberg unit contain proteasome. This proteasome formation by the substrate combine, and in this way the lysine side will be separate and the peptides and amino acid will be separate, and the deubiquitination will lead to separate the ubiquitin, and this ubiquitin again recycling. This again ubiquitin again recycling, so the ubiquitin will again work, so it will not end product but it will create the end product of the protein which that is unrequired, so the lysine side will be separate. So anyhow, I hope you make sense about that. The deubiquitination due to the deubiquitinase enzyme will lead to separate the lysine from the ubiquitin. So in this way, the again recycling will occur. The cellular function of the ubiquitination is the proteasomal degradation and the selective autophagy, endocytosis, and the receptor trafficking. Remember about that thing. For example, the bacteria will invade into the cell. So remember, this basically is the unrequired protein. So in this, the bacteria will be the cell automatically autophagy will occur. While on the other hand, the cell signaling that like the necrotic factor kappa B and the damage response, for example, the DNA will damage and the cell will damage. So the ubiquitination will recover the damaged cell, and in this way or basically the programmed cell death and the cell cycle control for the control of the protein. For example, the excessive amount of the protein for the cell cycle lead to cause cancer. So in this way it will inhibit, and in this the ubiquitin will play important role for the cancer prevention. The normal and dysfunction we will discuss now. So first of all, we will understand about the normally. So the normal cellular function and homeostasis, the proteostasis. Yes, the proteostasis means the maintenance of the protein in our body, like the bad protein and dysfunctional protein will remove the normal signaling and the cell survival. The next one is the cellular dysfunction and the disease. So in this is a developmental disease like cancer and developmental disease will progress. So the target drug is important for the dysfunction prevention due to the ubiquitin use, and the neurodegenerative proteins and the neurodegenerative prevents protein and the aging prevention can be occurred due to this. I hope you make sense about the ubiquitination and de-ubiquitination. Thanks for watching. See you in the next video.